Hello and welcome. My name is Logan McBride, and today I will be demonstrating my understanding of meiosis by sharing this slide to you. Meiosis is the process that occurs in the sex cells, sperm and egg cells, of an organism. Meiosis has two sets of 5 stages. Today we are going to talk about all of them. So let's begin. To begin, interphase 1 is the first phase of meiosis. In this phase, diploid germ cells, which are the reproductive cells, their chromosomes replicate. As you can see here, there's 4 chromosomes, and they have replicated to create sister chromatids. In prophase 1, the chromosomes begin to thicken, the nucleus disappears, and the spindle fibers begin to form. Also in prophase 1, tetrads are formed. Tetrads are when 2 sister chromatids, maternal and paternal, join together to create homologous pairs. This is important because it sets the building blocks for our next topic, crossing over. Crossing over also occurs in prophase 1. Crossing over is the exchange of genes between homologous chromosomes. There are many possibilities of crossing over, as you can see in this photo. Crossing over is important because it is one of the 3 main components of reproduction that can help in the genetic variation of an organism. Moving on, in metaphase 1, the chromosomes begin to line up along the cell equator. Each homologous pair becomes attached to a spindle fiber at its centromere right here, as they prepare to be pulled apart for anaphase 1. Anaphase 1 is when the spindle fibers begin to contract, separating the sister chromatids. As this is happening, the sister chromatids are pulled to separate sides of the cell. Also in anaphase 1 is when random assortment occurs. Random assortment is the way chromosomes get organized into gametes, which are sex cells. Specifically, how they randomly become assorted. This means that sex cells will have different combinations of chromosomes. Random assortment is important for the same reason as crossing over. It creates genetic variation in the reproduced organisms. Random assortment is the second of the 3 main contributors to genetic variation. In telophase 1, the first cytokinesis of the meiosis process begins. In this cytokinesis, 4 sister chromatids, in this picture, are split evenly into 2 cells. This is important because we have now gone from a diploid cell to 2 haploid cells. This is the end of meiosis 1. Meiosis 2 is often related to that of mitosis due to the fact that they are very similar in their phases. Let's get into prophase 2. In prophase 2, the chromosomes begin to thicken, the nucleus disappears, and the spindle fibers begin to form. In metaphase 2, the sister chromatids this time, not homologous pairs, line up on the cell equator as the spindle fibers attach to each other chromosome at the centromere. In anaphase 2, the sister chromatids of the 2 cells are pulled apart by the contracting spindle fiber, separating the chromosomes to opposite poles to prepare for telophase 2. Telophase 2 is when the second cytokinesis occurs. In this cytokinesis, you can see that we now have 4 new haploid cells. These cells are what are known as sex cells, the sperm and eggs. As I explained in telophase 2, we now have 4 haploid cells, the sex cells. These cells can now be used to reproduce in species that conduct in sexual reproduction. When the 2 types of cells meet, it is called fertilization. Fertilization is the 3rd component of 3 that contributes to the genetic variability of an organism. With 200+ million sperm cells released during reproduction, the possibilities of cell makeup are unthinkable. After explaining all the phases of meiosis, let me recap and note the importance of meiosis in reproduction. Unlike mitosis, meiosis's resulting cells are haploid to their parents, not diploid. This is important because when the 2 sex cells meet, sperm and egg, the cells haploid, so when added together they create an embryo, later to be an organism of the same species. The 2 haploid cells create a diploid cell, matching the chromosome count of those of the parent. Without this, the organism would not be the same species. For example, humans have 23 pairs of chromosomes. This of course equals 46. Human sex cells have 23 chromosomes. When they meet, 23 plus 23 equals 46, matching that of the amount of the parent somatic cells, aka body cells. If sex cells were to be diploid and have 46 in humans, 46 plus 46 equals 92. Therefore, no longer the stomatic body cells of the parents match the offspring. Meiosis is also important because, unlike mitosis, it creates genetic variability. In mitosis, the resulting daughter cells are identical to the parent cell. In meiosis, the daughter cells can have infinite possibilities, not identical to the parent cell. Being variable is important because if every family line continues to share identical cells as generations go on, and one virus or bacteria or illness is deadly to them, it would wipe out their whole family. With genetic variability, organisms can still struggle against diseases, yes, but other organisms of the same species can fight off those diseases or even be immune to them. This is why meiosis is extremely important in sexual reproduction and genetic variability. To conclude my video today, this has been my explanation of meiosis, its importance, and its phases. With this being said, my name is Logan McBride, and this was my demonstration of my understanding of meiosis.